This is the third and final video in a series of 3 looking at the detail of what happens during photosynthesis. And in this video we look at stage 2 of photosynthesis in some detail, the light-independent reactions. In the last video we considered how the first stage of photosynthesis, the light-dependent reactions, take place in the thylakoids and use some fairly sophisticated protein structures embedded in the thylakoid membrane. For stage 2 of photosynthesis, known as the light-independent reactions, we move outside the thylakoids into the stroma. The stroma is a space inside the chloroplast filled with an aqueous solution. So we're going to be looking at a series of reactions that take place in solution in the stroma. In the last video, in looking at stage 1 of photosynthesis, the light-dependent reactions, we explored how those reactions produce 2 very useful substances, ATP and NADPH. And now in this video, looking at stage 2, the light-dependent reactions, we'll see how those are used to take inorganic carbon as carbon dioxide and fix it, put it into organic molecules that are useful to the plant and other living things. This series of reactions is called the Calvin cycle, or more fully the Calvin-Benson-Bassam cycle. Can look a bit intimidating when you see a diagram of the whole lot at once, like the one shown here, but don't worry, we'll look at it step by step, consider some of the detail, explain what's going on and make sense of it. It's called a cycle because there is an extent to which it goes round and round and round, as you can see that in the diagram there, but at the same time we can think of it having a start and an end. The start really where carbon dioxide is fed in, and the end where we make the useful stuff that is the purpose of photosynthesis, which will turn out not to be glucose, despite what the simple equation for photosynthesis might suggest. It's actually a substance called glyceraldehyde 3-phosphate, or G3P for short. And this is a very flexibly useful substance for the plant. It can use it to synthesize a whole range of substances, certainly glucose, but also starch, cellulose, other carbohydrates, and even other types of molecule entirely. Before we consider the reactions that take place, we're just going to look at a molecule that's really a key player in this whole cycle, and so is found in large amounts in the stroma. It's the molecule that carbon dioxide gets joined to at the start to turn it into an organic form of carbon, and its name is ribulose 1,5-bisphosphate, or RUBP for short. Its structure is shown here. The carbons are the black circles, so it's a 5-carbon chain. At each end there's a phosphate group, so that's a pink phosphorus atom surrounded by some oxygens, and then along the chain there's various hydrogens and oxygen containing groups. So this substance really is fundamental to the Calvin cycle, and we'll see how it first picks up carbon dioxide. A few processes happen to turn it into the G3P, and then it gets regenerated, ready to go around the Calvin cycle again and again. So the first step that we're going to consider now is where carbon dioxide is used in photosynthesis. It's known as carbon fixation, and it directly involves the substance we've just mentioned, the ribulose bisphosphate. Let's look at the detail of what happens in this first step. So the equation here shows the structures of the substances involved in a rather more conventional chemistry way, showing the symbols for the different atoms. And we can see how one molecule of ribulose 1,5-bisphosphate reacts with a water molecule and a carbon dioxide molecule to make these 2 smaller molecules of 3-phosphoglycerate, or 3PG for short. Each of those small product molecules has 3 carbons in. So what we've done, we've taken the 5-carbon chain of the RUBP, added an extra carbon from the carbon dioxide to make 6, and then this is immediately split into 2 smaller 3-carbon molecules, each of them with a phosphate group at one end. The reaction doesn't happen spontaneously, doesn't happen on its own. So in order to get the water and the carbon dioxide to react with the RUBP, we need an enzyme, a biological catalyst. And in this case, it has the rather fancy name ribulose 1,5-bisphosphate carboxylase, but is more commonly known as Rubisco. Rubisco is generally taken to be the most abundant enzyme on Earth. Scientists estimate that at any one moment in time there's about 40 billion tons of it around. And if you think about it, that's not surprising because it's to be found in the chloroplasts in pretty much every plant cell and algal cell and so on. And it's really fundamental to life as we know it because it's this enzyme that allows the photosynthesizers to fix carbon dioxide, the inorganic form of carbon from the atmosphere, into the useful organic substances that living things really depend on. So the first step of the Calvin cycle left us with these 3-carbon 3-phosphoglycerate molecules. And we're now going to look at how the ATP produced in the light-dependent reactions comes into play to take those molecules onwards on the journey towards the G3P photosynthesis product. So this second step in the Calvin cycle is known as a phosphorylation reaction, and that's because it adds a phosphate group to our 3-phosphoglycerate molecules. So you can see in the equation here, each of the 3-phosphoglycerate molecules produced reacts with a molecule of ATP and gains a second phospho group to its structure. So it turns into 1,3-bisphosphoglycerate, or 1,3-BPG for short. The ATP, adenosine triphosphate, in the process it loses one of its phosphate groups and so is converted to ADP. And of course that's going to find its way back to the thylakoid where it can be converted back into ATP at the ATP synthase, as we looked at in the last video with the stage 1 reactions. Just as in the first step of the Calvin cycle, this reaction needs an enzyme to make it happen. And so in this case, the enzyme is known as phosphoglycerate kinase. The third step of the Calvin cycle can in some ways be considered to be the end of the process of photosynthesis, because it's this step that's going to produce the useful G3P that is the final product of photosynthesis as far as we're concerned. So it's a type of reaction known as reduction, and we'll look now at the detail of what goes on in this step. The equation here shows what's going on in this third step of the Calvin cycle. Each of our 1,3-bisphosphoglycerate molecules produced in the previous step gets converted into a molecule of glyceraldehyde 3-phosphate, otherwise known as G3P. You can see that essential to this process is the NADPH that was produced in the final step of the light-dependent reactions. So it's at this point that substance gets used. During the course of the reaction, the 1,3-bisphosphoglycerates lose one of their phosphate groups. You can see in the product there's now just one phosphate group attached. So another product of this reaction is a phosphate ion, represented as Pi in the equation. Along the way, the NADPH gets turned into NADP+, and that's going to make its way back to the thylakoid, where it can be regenerated as NADPH by the light-dependent reactions. As with the previous two steps in the Calvin cycle, we need an enzyme for this reaction to happen, and so here we meet a third enzyme, glyceraldehyde 3-phosphate dehydrogenase. So that's the enzyme required for this step. And at this point you can pat yourself on the back if you've been following the videos through and keeping up, because we've now made the precious product of photosynthesis, the G3P, that's then going to be available to the plant to turn into glucose and all sorts of other useful substances that the plant depends on, and also that we depend on as the original source of the food that we eat. Our story's not quite over though, because if we left it here, the Calvin cycle wouldn't be a cycle at all. The ribulose 1,5-bisphosphate that was used at the start, the RUBP, would have been consumed, and so for any further action in the Calvin cycle, a source of fresh RUBP would be needed. So we're going to just look at the detail of how the RUBP is regenerated And what we'll find actually is that most of the G3P made in the Calvin cycle is not available for the plant to turn into glucose and other substances, but gets used up regenerating the RUBP. So the diagram here shows a rather symbolic representation of how RUBP is regenerated rather. Each pass through the Calvin cycle uses one RUBP molecule and produces two G3P. So what we're looking at here in a way is the result of 3 cycles round the Calvin cycle. 3 RUBPs would have been used up, reacting with a carbon dioxide each, and 6 G3Ps would have been made. Each pass through the Calvin cycle produces 2 G3Ps. And so the diagram showing here is that for every 6 G3Ps made, 5 of them go to regenerate the 3 RUBPs that were needed to produce those 6 G3Ps, and only 1 of them is available for the plant to turn into the glucose and other useful substances required. It's not something that just happens spontaneously. There's other substances involved. You can see from the equation that it uses a couple of water molecules. It also uses a further 3 ATPs, so those would have been made by the light-dependent reactions earlier on. As a result of these reactions, we regenerate 3 molecules of RUBP for use again in the Calvin cycle, and we also produce 3 ADPs that can head back to the thylakoid and be regenerated as ATP by the light-dependent reactions. We also get a couple of phosphate ions. There's several steps really involved in this regeneration. It's quite a complicated process, but you don't need to know the details of that. So we're not going to cover it here. You can research more about that online if you would like. And as before, enzymes are involved, and one of the main ones is called phosphorybulokinase. So we now have sort of reset the Calvin cycle with the RUBP ready to fix more carbon dioxide, and so over time there's a steady production of G3P as you pass around the Calvin cycle, with the RUBP being regenerated each time ready to be used again. If we crunch all of those steps of the Calvin cycle into one overall equation, we get what's shown here. This is really for 3 passes round the Calvin cycle. So 3 carbon dioxide molecules will have been fixed, using, to do that, 5 water molecules, 9 ATP molecules, and 6 NADPH molecules. And for all of that material, we produce our 1 useful G3P, the product of photosynthesis. We're also going to get the 9 ADPs and 6 ADP NADP pluses to be regenerated by the light-dependent reactions. We get 8 phosphate ions and 2 hydrogen ions as well. If you want, you could go back and look at the steps of the Calvin cycle and do the maths and see how those 3 passes through it do indeed require that number of ATPs and NADPHs. The process of turning G3P on into useful products would be a whole nother topic of study beyond this, so we won't look at that here, but just suffice to say each of those G3Ps has 3 carbons in it. Glucose is a 6 carbon molecule, so to make 1 glucose you're going to need 2 G3Ps, which of course means 6 carbon dioxide molecules reacting originally in the Calvin cycle, so 6 passes round the Calvin cycle. So that would be in a way double what was shown in the overall equation on the previous slide. So this brings us to the end of our journey through photosynthesis. As you hopefully have picked up, it's a long and complicated journey. It started in the I suppose it started in a way with the roots and the stomata bringing in the raw materials and the photons of light, but then there were the processes in the thylakoid membrane using light energy to generate those ATP and NADPH molecules, and then those feeding out into the stroma where the carbon from carbon dioxide was then fixed and processed into G3P. And along the way, the plant has very efficiently recycled everything. The ATPs, the NADPH being continuously produced from ADP and NADP plus, and then turned back into those things. The RUBP, the ribulose 1,5 bisphosphate, being used to capture a carbon dioxide molecule and then turned back into RUBP once again. So this is a continuous process that can cycle round and round and round, consuming water and carbon dioxide, using the sun's energy, and churning out G3P as a production line, which then life as we know it is built on.